scientists need to use microscopes to study cells. There are different kinds of microscopes, and today we're going to introduce you to the types you need to know about for your A levels, which are the electron microscopes, which we've already seen some images from, and the light microscope or optical microscope that you're used to using at school and will again be using with us at college. So you'll be following this video with your keynotes, pages 13, 14 and 15. And we're starting on page 13 with the optical microscope, the light microscope. So what do you need to know about this wonderful light microscope, the one that you're going to use in the laboratory? Well, it allows us to view specimens illuminated with light, which then is focused using a number of glass lenses. Okay, and you view it either with your eye or photographic film, or it can obviously go straight to a computer now as well. And that allows you to select different lenses to magnify to different magnifications the specimen that you view on the stage here underneath your lens. So we've used them, we're used to using them hopefully, we will do again. We do need to know some of the features of them. Okay, so one of the wonderful things about the optical microscope is that the specimens that we view can be alive. Okay, so they can be living specimens, for example, single cell organisms. We can observe their behaviour, how cells interact, for example, how they move as they're a living sample. But we can also fix the sample at a particular stage in its development and view it as a dead sample, as you will be doing. When we do our root tip squash, you will view the root tissue of a garlic plant during, hopefully, cell division. So we'll kind of capture the cells right in the middle of doing that process, and we'll kill them, fix them and stain them. And that takes me to the other thing. So although we can see colours, because obviously we're using light to view the specimen, often we might have to enhance our samples. Sometimes we add coloured stains, dyes, and often we stain for things like DNA. So in these two diagrams, this first one here, this isn't stained at all. These are the chlorophyll. This green is the chlorophyll of these chloroplasts under the microscope. So this is viewed by the optical microscope. These are plant cells, and because of the green pigment, we can see them already, those chloroplasts. Whereas in these two pictures here, these are animal cells. Oh, sorry, this is an animal cell and this is a plant cell, and both of them have been stained this time with a type of stain that stains DNA. So you can see that the nuclei have stained very strongly. Okay. So what else do you need to know about the light microscope? Well, really, it's the limitation of the light microscope that we really focus on now, okay? Because it is limited in its magnification to about 1,500, which doesn't allow us to magnify samples to a high enough magnification to observe the small structures within them. So if you think about the cells that we were taught about at GCSE without all those additional organelles, the Golgi apparatus, the rough endoplasmic reticulum, etc., well, we can't actually view any of those structures down the optical microscope. So it required the invention of a new type of microscope to be able to give us information about those structures that we're unable to observe with our optical microscope, our light microscope. So really what it only allows us to do is to observe individual cells, so the shape of cells and how they fit together in tissues, and also some large structures like the nucleus. Some of the better light microscopes that might use blue light, they can observe some other structures, maybe the bigger ones like we saw in the previous one. You can see the chloroplast. Some optical microscopes can be quite good and see larger structures like the chloroplast in a little bit more detail, not quite as fuzzy. but you're really restricted to very large structures inside cells being visible. Nothing like the intricate detail that we've been learning about recently. So why is that? Well, this is probably the most important learning point for your limitations of your optical microscope. The magnification could be increased. We could use bigger lenses to magnify the object, the specimen, to a greater amount. But unfortunately, because the wavelength of light is quite long, it doesn't matter if we magnify further because the resolution would not improve. Okay, so what's resolution? So if magnification is how much you sort of increase the size of some of the specimen as you view it, what's resolution? Well, resolution is the ability to see two separate objects as separate. Okay, now the resolution of a light microscope is limited by the wavelength of light. So if we consider two objects, then the light microscope, if those objects are less than half the wavelength of light apart, okay, they'll be seen actually as one blurred object. So one object will be visible using the light microscope. Whereas if objects are far enough apart, okay, so they're more than half a wavelength of light apart, they'll be viewed as separate objects using the light microscope. So we say that the resolution of the light microscope is limited by the wavelength of light, and that means that we can't magnify any further because the resolution is what is limiting our ability to see those smaller objects as separate objects and not just one thing. Okay, so that meant we needed to invent a new type of microscope, one with a better resolution that would allow us to visualize objects that are closer together and so are smaller. And that would allow us a greater magnification. And that came with the invention of the electron microscope. Now this is different in that it uses a beam of electrons rather than light to illuminate the specimen. Okay, I know this may seem strange, but electrons can be forced into a beam, and when they do, they behave as a wave. Okay, and they can be produced really easily. You basically use a hot wire. So a beam of electrons is produced and is passed through the sample, okay, focused by electron magnets. And then there is a film or a photographic film that detects the electrons as they pass through the sample, and that produces an image that can be interpreted by scientists. Okay, so our electron microscope then, because we've got a beam of electrons now, it's got a very small wavelength, much smaller than the wavelength of light, which means that it is able to produce images that observe objects as small as ribosomes, 20 nanometers. So now we've got a much smaller wavelength to our electron beam, and that means the resolving power of the microscope is much higher. Okay, so it's got a greater resolution. That means we can take it now to greater magnifications, and still at those higher magnifications, the image will show objects as separate because the resolution is higher. So in terms of resolving power, the electron microscope's resolution is 1 nanometer. Okay, so comparing that then to the optical microscope, you can see it's got a much higher resolving power because the distance between the two objects must be, to be resolved as two separate objects, is 0.5 nanometer. which means that its effective magnification is up to 500,000. And just to illustrate that to you, look at this electron micrograph here. You can see not only our ribosome made up of two sub units and lots of ribosomes, you can actually see, make out the mRNA and the proteins forming. So that's how high the magnification is. So to do that though, we need to now start to think about some of the limitations of the electron microscope. Okay, so to be able to pass an electron beam through the sample has got to be incredibly thin. So you can see this kit here that is used to produce our specimens for viewing down the electron microscope. We first have to fix the specimen in a wax or resin, and then we've got to cut a very thin section through that sample to be mounted then to view down the electron microscope. It's also got to be viewed, that specimen, in a vacuum, and that's because the electron beam would be easily scattered by the molecules in air. So we have to take out all the molecules and leave it a completely empty space, which is a vacuum. And that means the electron beam will then pass straight through that vacuum and through the sample. So anything you view down the electron microscope, because the specimen is fixed in this way and because it's viewed in a vacuum, must be dead. And that's a real limitation to what we view down the electron microscope. Okay, and also that processing, you know, the electron beam itself, the sometimes addition of electron dense chemicals that are used, and that slicing means that sometimes we produce artifacts. Now, artifacts are things that you view in your sample that are the result of preparing your sample for the electron microscope and not for as a result of the electron microscope itself. There's a lovely little video from a scientist who prepares samples for use under the electron microscope at the link at the bottom, so you can learn a little bit more about those artifacts and how that happens. So this means that samples are dead and we can only produce black and white images because we're now using an electron beam, and also there's the potential that we might produce artifacts, so things we can see that are not really in that cell or not really in that tissue, but are structures we observe because of the process of preparing the sample. Okay, so thinking about our optical microscope compared to our light microscope, they are much cheaper. Okay, so I wouldn't use that as a mark point particularly, but they are much cheaper, and you don't really need someone to be specifically trained, or you don't require a lot of training to use them. Whereas electron microscopes, as you'll see from that video if you watch it, the people who use them are highly trained, highly skilled, and they're very expensive bits of kit. So even with complex staining procedures, the electron microscope can only produce black and white images. So here's our chlorophyll as chlorophyll stained, if you like, chloroplasts, that when viewed down the optical microscope, we can see the colour of the pigment that's actually naturally there. Although we can, because of the high resolution of the electron microscope, we can zoom right in on one of those chloroplasts to those granum, those stacks of thylakoid discs, but what we can't see is the colour of them, okay, which we can see down the optical microscope. Okay, so we're preparing for the question at the end of the paper, which is contrast how an optical microscope and a transmission electron microscope work, and contrast the limitations of their use when studying cells for 5 marks. So this is the kind of question that would be at the back of paper 1. Okay, so the first thing you need to do is to copy down the answer, mark it, and then I'm going to ask you to improve the student's response. Okay, so if you pause the video here and complete that task, then rejoin me to go through. Okay, so I'm going to go through this answer note, and I'm going to first start by marking this particular answer. Okay, so it says that the transmission electron microscope uses a beam of electrons that are focused by electromagnets, which is true. Okay, but in the question we're asked to contrast the transmission electron microscope with the optical microscope, and you can see that this student here has not gone on to do that. So although it looks like they've said something that is correct, which they haven't then contrasted it. So they haven't said the light microscope uses a beam of light, which is focused by lenses. So they can't have a mark point here. Okay, they are short of detail for the contrast. So they didn't show the other side. This beam has a short wavelength, giving the transmission electron microscope a much better resolution. Now I'm just going to circle that word for you, and so a higher functional magnification. Again, we've not done anything here to contrast limitations, because the limitation is of the light microscope. So we really needed to say the light microscope's got a lower resolution, okay, and so a lower magnification to get that mark point. And of course that's because the wavelength of light is longer. I'm just going to give us a little star here, because this better or poorer, we have to be really careful. We used to be able to use better and poorer, but actually on our new exam board, on the new spec, they want you to say how it's better. So just be really careful not to use that, and to use higher or lower. So you're actually saying which is the better. So a higher resolution is better. Okay, so still no marks because we haven't got the contrasting detail. So the transmission electron microscope's got, so with it you get greater detail, that the structures of organelles can be seen that are not visible using the light microscope. So now this is the only time when they have done a contrast, okay? And I think saying greater detail at the structures inside in organelles being seen by the electron microscope that are not visible, so that's if you like, a limitation of the optical microscope. Now they can have 1 mark. So you can see that when they're aiming for 5 marks, we really need almost a page of A4 of compare and contrast. So making sure you do the electron microscope and then follow that by the optical microscope. Okay, so having a look at the additions that you should have made. So to get that mark point out of the first line, we wanted to contrast how they work. So they use a beam of electrons for the transmission electron microscope, whereas for the optical microscope, a beam of light. So there's actually a mark point for that, and a second mark point for what's focused. So in the transmission electron microscope, the beam of electrons was focused by electron magnets, whereas it was lenses for the optical microscope. Get a mark point there as well. If it was compare and contrast, if we're looking for similarities, both the transmission electron microscope and the optical microscope do pass this beam through the sample, and that's something I'll come back to in a minute. But that, of course, is not in this particular answer. So we've got 2 mark points out of that first line now. Let's see what we could do to the next one. The electron beam has a shorter wavelength than a light beam, so we could do in a contrast, giving the transmission electron microscope a much higher, so you see that improvement there with the higher resolution, and so a higher functional magnification than the optical microscope. Better now, which is limited in its resolution by the longer wavelength of the light waves. Okay, so now we've got that limitation because the longer wavelength of the light waves is limiting the resolution. Okay, so in other words, we've got a lower resolution. So the transmission electron microscopes, greater detail, like the structures inside organelles can be seen. Just to improve that a little bit, that are not seen using the optical microscope, giving an example like the crista of the mitochondria. But that was our original mark point. And then there's some extra material they've not even touched on. Okay, so the transmission electron microscope can only be used to view dead material, as the specimen is viewed in a vacuum, whereas the optical microscope can be used to observe living cells. Now, the detail of vacuum had to be here for this mark point, so it wasn't just dead versus living, you had to explain why. The optical microscope produces colour images, where the transmission electron microscope can only produce black and white images. Will also get you a mark point. And the transmission electron microscope preparation may create artifacts, whereas the optical microscope preparation is simple and unlikely to damage the material being viewed. So this is much more likely with the complex cutting of the thin samples and in the resin and the staining techniques that are used and the electron beam itself. That's what creates artifacts. You don't get artifacts in the optical microscope, and any damage you would do to the tissue, it is much less because obviously it's a very simple preparation. Okay, so they've got their 5 marks now. Okay, and look how hard they had to work to get them. So have a look at your answer, make sure you've got all that detail. That is a very commonly asked question at the end of paper 1. Okay, so you have got a table summary in your keynotes, but just learning that table is not going to be enough. Can you see how the question asks you to use that information, and you do need to put the detail in? Okay, so you might want to take a picture of this and pop this into your keynotes as well, or add additional notes into your keynotes. Okay, so the final thing we need to know about microscopes is that there are more than one type of electron microscope. So we've been talking there about the transmission electron microscope. So the transmission electron microscope is one where the electron beam is passed through the sample, just like in the optical microscope, the light passes through the sample. But that's not the only way we can use an electron microscope. Okay, so we'll just review our transmission electron microscope first of all. So it works in much the same way as the optical microscope. You transmit your beam through a thin sample. Okay, of course it has to be a lot thinner in the case of the electron microscope than the optical microscope because it's an electron beam. And then we focus those electrons on the image using our electromagnets, and the image is produced on screen or film. It's the most common form of electron microscope, and the reason is it gives us this wonderful high. Now I've changed this for your keynotes just now. I would like you to do the same thing there. Make this a higher resolution rather than greater. It is higher than the scanning electron microscope's resolution. Okay, but there is one limitation to the transmission electron microscope that you can overcome with our next type, and that is that the image is two-dimensional. So you're seeing a cut through of a cell or a cut through of a structure like an organelle, which of course gives us lots of interesting information about the structures inside that cell, inside that organelle. But sometimes to get the understanding of something, it might be better to have a 3D image, like the surface of a structure, for example. So if you have a look at these finger-like projections, these are the microvilli. Quite difficult to get the idea from the transmission electron microscope that these are finger-like projections from the surface of these epithelial cells. Can you see that sometimes they're caught in transverse section and sometimes in longitudinal section? They don't look like complete finger-like projections. When viewed by the scanning electron microscope, just the image of these cells, I think you get a real clear sense of those microvilli, those long finger-like projections of the cell membrane from the surface. So how is that image produced? Well, the scanning electron microscope, this time you place your beam, if you like, at an angle onto the surface. Okay, so it's the fine beam is sort of bounced off the surface and scatters the electrons. Okay, so it does give you a lower resolution, but it gives you excellent 3D images. And also, you don't have to have thin samples, so you can get external sort of 3D images of complex organs, like, for example, the eye of a fly and those kind of things, are kind of famous image examples. Okay, so here is just a little bit of, just explain a little bit more about that scanning electron microscope. Okay, so this time we're not passing the electron beam through the sample, we are scattering it off the surface, and then the electrons that have been scattered have been detected, and that's what forms this three-dimensional image. Okay, so that's useful when studying the surfaces of things, because obviously we get more of a sense of the three-dimensional structure, like this ant's head, or these pollen grains. I do like the pollen grains, they're nice. So again, lovely three-dimensional images. So the exam board is really keen that you first of all know the limitations of each type of microscopes and the benefits of each, and know generally how they work. Okay, so again, that information is found in your keynotes from page 13 to 15. You do need to construct your answers though, so do make sure that you don't just learn off by heart. You need to be ready to apply it to what the examiner is asking you. So here is one final example. If you are given a picture in the exam and asked what kind of microscope, then you would have to use that information and apply it to your answer. Okay, so here we've got a picture of a mitochondria. We can see the inner membrane, we can see the matrix space, we can see the crista, the folds of the inner membrane and the outer membrane, and within an animal cell. So straight away, you would be thinking, I'm hoping, that this is an electron microscope image, okay? But you need to give them evidence. Now, what if this was 4 marks? What 4 pieces of evidence could you give? Well, first of all, that the electron microscope's got a higher resolution, and so a higher effective magnification, allowing the image produced to show the crista of the mitochondria, because that's an internal structure of a very small object. Okay, so that would be my first piece of evidence. Now, it's a 2-dimensional image, which tells me again that this is the transmission electron microscope. So because I've passed my beam of electrons through the sample. It's a black and white image, so again that confirms it's an electron micrograph. So it's picking out things that give you evidence. Okay,